Hi, I am Niranjan Boisak. I am a sugarcane molecular biologist at the Louisiana State University Agricultural Center, housed at the School of Plant, Environmental and Soil Sciences, Baton Rouge, Louisiana. Today, I take advantage of this opportunity of the virtual field tour to demonstrate some of the work that we do here at the Sugarcane Molecular Genetics Lab. First of all, I would like to thank the stakeholders for supporting this molecular research through funding by the American Sugarcane League. Many of you growers, producers and processors are not aware probably about what we do here at the Molecular Genetics Lab. So, this is an opportunity for me to showcase some of our research. So, what we do? Our main goal is actually to devise molecular tools to complement sugarcane breeding program in the development of improved sugarcane varieties. And how do we accomplish that? So, we have different ways that we utilize these molecular tools. So, number one is actually when we started working in the molecular genetics program of sugarcane, the first thing we wanted to do is to look at the genetic diversity that is present among all the sugarcane clones that are existing in Louisiana. So, we use the molecular tools to identify the genetic diversity, and what it does, it gives us the information that how diverse the parents or the clones that we have in Louisiana sugarcane industry. And we pass on that information to the sugarcane breeders who can make informed crosses to produce the offsprings that would have more hybrid vigor. But we know the more diverse genetically the parents are, the more possibility of vigorous the progeny are. Another opportunity that we use or we take through this molecular research is to identify DNA markers that are linked with a particular trait of interest. For example, high sucrose, disease resistance, higher cane tonnage. So, DNA markers are very faithfully inherited across generations. They are not influenced by the environment like your trait does. So, when we select for a particular trait by using the DNA markers, the selection efficiency is higher, because selection based on just what we see in the field can sometimes be erroneous, because maybe the environmental condition was not conducive to allow the expression of the disease or that particular trait. So, in that way DNA markers are very handy. For example, we have been using the DNA markers that are linked with brown rust resistance in Louisiana sugarcane breeding program starting from 2014. And we started in 2014, the brown rust gene, the frequency was only 5% among this Louisiana sugarcane germplasm. But using the molecular markers or the DNA markers linked with that gene, we could improve the brown rust resistance gene frequency up to 25% as of 2020. The brown rust resistance gene that I just mentioned was actually identified by a group from France in collaboration with other international scientists. It took them almost 12 years to identify that gene. With your funding here at the American Sugarcane League and with the United States Department of Agriculture, we were able to identify genes for leaf scald resistance, and we identify the markers that are linked with those genes, and we have been using these markers for leaf scald resistant screening of our parents and offsprings from 2020 onwards. So, this is actually a great achievement in terms of the molecular genetics research. There is another opportunity or another advantage of these molecular tools is genomic selection that we have been just recently starting to work on. So, what genomic selection does, that we do take the phenotypic and the pedigree information of all the Louisiana sugarcane parents, elite clones, commercials that are in the Louisiana sugarcane industry, and we combine that data with the high throughput genotyping data and then employ statistics tools. They are very sophisticated statistical tools, and by that way we can actually identify genetically superior offsprings that have the gene combinations from the parents, yet can supersede both the parents in their performance. So, this is genomic selection, and as I told you, these molecular research involves a lot of work, starting from DNA extraction. So, we isolate the DNA from just a tiny bit leaf tissue that we collect from the field or from the crossing cart or from the greenhouse, and then isolate the genomic DNA and then use PCR that we call polymerase chain reaction, and put the PCR into this sophisticated equipment where we can actually see the DNA fragments in a gel or in some other equipment that we have recently purchased through the funding from the American Sugarcane League. So, what we are looking at here is actually the brown rust resistance gene that is present or absent in the Louisiana sugarcane clones. So, the red ones, the red dots over there represent the clones that contain the brown rust resistance gene, and the blue one, they do not have that gene. So, with that, I would like to again thank the American Sugarcane League and you, all the stakeholders, for supporting our research. As you can see, the molecular genetics research has a tremendous potential, and it will have far reaching benefit to the Louisiana sugarcane industry moving forward. Thank you.